These are the recorded lectures for cellular structure. Let's see, let me get the slideshow going. Okay, the chapter begins with a little bit about microscopes, and that is because looking at cells, individual cells, involves microscopes. A microscope is an instrument that magnifies an object. The light microscope is the one that we use in biology lab, and it uses light to illuminate a specimen. A light microscope can magnify a specimen up to 1,000 times. But then, if you really want to see detail on viruses and bacteria, you need an electron microscope, which we do not have at Wilson Community College. It's really expensive, but it gives you much higher magnification resolution, which means it's finer. It's just more clear picture and more detail. So this is a bacteria, an image of, well, actually you can see more than just one, but this is an image of bacteria with an electron microscope. All right, most cells are too small to see without the aid of a microscope. Within cells, there are numerous structures that also need microscopes, especially electron microscopes. Now remember, there are two types of cells. Eukaryotic cells are larger than prokaryotic cells. And eukaryotic cells have a nucleus and other tiny organs. They're not really organs, but they're tiny structures that act like organs called organelles that have specialized structures and functions. So we're going to learn about the various organelles within an animal cell, a eukaryotic animal cell, and a eukaryotic plant cell. So first, the largest organelle is the nucleus. And the nucleus is surrounded by the nuclear envelope or the nuclear membrane, which is a double membrane. And it has pores which allow molecules like RNA to move out of the nucleus. DNA inside the nucleus has formed into a material called chromatin, which is where the DNA has wrapped around these proteins called histone proteins. But chromatin is still a very uncondensed or spread out, messy form of DNA, I guess you could say. When the cell is about to divide, the chromatin coils up into chromosomes, which are rod-shaped structures. And humans have 46 chromosomes in all their cells. Another structure you'll find in the nucleus is the nucleolus, which is the job of the nucleolus is to form ribosomes. And ribosomes, are they on here? Are not on here, but ribosomes synthesize proteins. A peroxisome is a vesicle, small round vesicle, that breaks down waste, particularly fatty acids. The endoplasmic reticulum is this network of channels. You see, most of them have these little red dots on them, and those little red dots represent ribosomes. So the rough endoplasmic reticulum contains ribosomes and is associated with synthesizing proteins. The smooth endoplasmic reticulum does not contain ribosomes, and it is associated with producing lipids and carbohydrates. And the cytoskeleton of the cell involves microtubules, which are involved in cell division and help maintain the cell shape. The microtubules are the largest of the fibers that form the cytoskeleton. The centrosome is formed of microtubules. and it's like these two bundles of microtubules that are perpendicular to each other. The centrosome is going to be involved in cell division. Intermediate filaments are medium thickness and they hold organelles in place. And microfilaments form the cell cortex, and cortex means the outside. So they line the outside of the cell right up close to the plasma membrane. The plasma membrane is the structure that encloses the cell and separates it from its surroundings. A lysosome is very similar to a peroxisome, but it digests food and waste materials. So a lysosome contains many digestive enzymes to help digest food and worn out cells and different things like that. The Golgi apparatus modifies and packages proteins and other molecules that are produced by the endoplasmic reticulum. The cytoplasm is everything inside the cell besides the nucleus, and the mitochondria produces energy in the form of ATP. A vacuole is a storage molecule. It's larger than a vesicle, and whereas a vesicle is small and round, a vacuole can have a like kind of shapeless, it won't have a fixed shape to it. And a plant cell, we see that there is a plasma membrane and cytoplasm, just like the animal cell. That there are three things that the animal cell did not have. There is a large central vacuole filled with water. There is a cell wall outside of the plasma membrane that helps maintain the cell shape. There are plasmodesmata, which are channels that connect the plant cells to each other and allow materials to flow throughout the plant from cell to cell. You still have rough and smooth ER, or endoplasmic reticulum. You still have a nucleus, nucleolus, and chromatin. You still have ribosomes. You still have a Golgi for modifying and packaging the products from the endoplasmic reticulum. You still have mitochondria for producing ATP energy, and you have peroxisomes for breaking down fatty acids and waste. But you have plastids, which store pigments, and this is found in plants and not animal cells. And the chloroplast in particular is found in plant cells and not animal cells, and that's where photosynthesis occurs. It stores the pigment chlorophyll as well as some other pigments. And then there's also the cytoskeleton. What you do not have, there is no centrosome or lysosome. Since plants make their own food, they don't have to do as much breaking down of their food because they make it already in a broken down form. So any digesting that occurs in a plant usually occurs in a vacuole or vesicle. They don't need a lysosome for that. The cytoplasm is different in eukaryotes and prokaryotes. In eukaryotic cells, the cytoplasm is everything between the nucleus and the plasma membrane. In prokaryotic cells, they don't have a nucleus, so the cytoplasm is everything inside the plasma membrane. The fluid, or the gel-like fluid inside of a cell, is called cytosol, and it's got ions, small molecules, and large molecules dissolved in it. The cytoskeleton is the fibers that we talked about, the microtubules, the intermediate fibers, and the microfilaments. And many metabolic reactions, including protein synthesis, take place in the cytoplasm of the cell. The nucleus houses the DNA and directs the synthesis of ribosomes and proteins. The nucleus contains the nucleolus, which produces ribosomes, and then ribosomes produce proteins. Chromatin is very, very unorganized DNA, and what chromatin does, chromatin is DNA wrapped around histone proteins. And what chromatin will do is coil up and condense into these chromosomes. There's one, and then here's another one. They're kind of like, chromosomes are like that. They're attached at the center. But condensed DNA is called chromosomes, and they come in pairs. So you can see what chromosomes look like here. They almost look like floppy, the letter X, but it's a floppy version of the letter X. But DNA takes the form of chromatin when the cell is resting, and it takes the form of chromosomes when it is in the process of dividing. The nucleolus, we already said, it says it aggregates the ribosomal RNA, so it basically produces produces ribosomes. And then what do ribosomes do? Ribosomes synthesize amino acids into proteins. They combine amino acids into proteins. The ribosome has a small subunit and a large subunit, and this is where the amino acids are attached. There's your protein. Mitochondria have two membranes, and the inner membrane is folded. Those folds are called cristae, and the reason for the folds is that the ATP, you can make a whole lot more ATP energy with a folded inner membrane than with just a plain flat one. Mitochondria does have its own DNA and its own ribosomes. Peroxisomes are small, round organelles. They carry out oxidation reactions that break down fatty acids and amino acids, and they can be used to detoxify poisons. Glyoxysomes are specialized peroxisomes in plants. I don't think we had that in the plant cell diagram. But they're responsible for converting stored fats into sugars. The endomembrane system is the part of the cell that's kind of like a factory. It begins with the nuclear envelope, or the nucleus itself, and then your rough ER, which synthesizes proteins, and then your, they don't have the smooth ER here, but the smooth ER would be outside the rough ER. And then the Golgi apparatus. The rough ER produces proteins because it has ribosomes attached, and ribosomes make proteins. And those proteins are modified and packaged into a transport vesicle that pinches off from the Golgi apparatus. So the endomembrane system modifies, packages, transports lipids and proteins. It includes the nuclear envelope. It includes lysosomes because lysosomes are made by this process. The rough endoplasmic reticulum will produce the enzymes, the digestive enzymes, which are proteins. And those enzymes will be packaged into a vesicle, and that vesicle is now called a lysosome. Vesicles or transport vesicles, the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane. If the Golgi packages up something that is designed to be sent out of the cell, it will fuse, that vesicle will fuse with the plasma membrane, open up, and then that product will be released outside of the cell. The endoplasmic reticulum is a series of interconnected membranous sacs and tubules. The rough ER modifies proteins, and it does contain ribosomes on its surface. The smooth ER synthesizes carbohydrates and lipids and steroid hormones, and also stores calcium ions, which is really important in muscle contraction and in the nervous system conducting impulses.